Hi, I'm Harith Akram. I'm a consultant neurosurgeon at the National Hospital for Neurology and Neurosurgery. I specialize in functional neurosurgery. I operate on patients with Parkinson's disease, and I do a procedure called Deep Brain Stimulation. During that procedure, we implant very thin electrodes deep in the brain. During this surgery, we use a very thin probe to guide how the electrodes get into the target. Now, when we remove that probe, there are often cells that are just attached to it naturally. And what happens to these cells is that they get discarded, which is a real shame because the information that we can get from testing the genetics of those cells can be very informative. It can help us understand better why Parkinson's disease develops, how it develops in certain patients, and how it responds to treatment. We get some genetic information from the brains of patients who have very generously donated them to science after their death. But the information that we get is not the same because when patients die, the natural cell degradation process starts to happen immediately. So we lose a lot of this information. At the National, we have this great opportunity to get these cells from every operation that we do, and then we send them to different labs, and we're very fortunate that we have a wide range of expertise around us, that we can all work together to understand Parkinson's disease better and help develop better treatments in future. The idea from this collaboration, which encompasses UCL labs as well as the Francis Crick Institute, is to get these cells and run DNA analysis, run RNA analysis, run protein analysis and protein assays, and look for inflammatory markers. All of this will then also get linked to imaging studies that we do routinely in those patients, and we can generate a three-dimensional atlas of all this information that will help us better understand the disease and develop newer treatments. This resource is also going to be invaluable to other researchers in the field. We wouldn't be able to do this without the support of the National Brain Appeal, and we're immensely grateful to them and to all the people who support the Innovation Fund. Thank you.